The overall goal of this study is to calibrate and test a novel device that can be used for in-situ measurements of biomarkers in cryospheric habitats. The device is based on laser-induced fluorescence emission technology, referred to as L.I.F.E. The current standard sampling methods lead to physical impacts on the specimen due to coring, disintegration of samples by chipping and sawing, and temperature shift by melting. These methods often lead to falsification of in-situ conditions. Therefore, a new methodology for the in-situ detection and characterization of microbial life is crucial. We have developed a novel, non-invasive, non-destructive method with a high spatial and temporal resolution. The application of a laser-induced fluorescence emission technique is based on the fact that supraglacial environments are inhabited, among other things, by phototrophic organisms. These organisms can be traced by the detection of fluorescent patterns of the porphyrin-based accessory pigments chlorophyll A and phycoerythrin, which are excited by a blue and green laser respectively. The portable dual wavelength kit weighs 4.5 kilograms and is used on a tripod in combination with an external computer. The lens tube is directed towards the specimen. Then, a green 5 mW laser hits the sample after passing a polarizing beam splitter that redirects polarized light towards the optical axes of the spectrometer. The specimen exhibits a fluorescent light, illustrated in red. Half of the collimated light passes the polarizing beam splitter and is focused through a servo-steered long-pass filter which removes the laser signals. Next, the signal hits an aperture slit which consists of two adjustable razor blades. A prism spectrally separates a fine line of light orthogonal to the slit aperture.